Okay, so this is part 5B. There is no separate notes for this, but if you look at the part 5A, you'll have the slides for part 5B. I just divided it up into two parts so that the lecture wasn't so long. Okay, so this is going to be still on adaptive immunity, and we're going to be talking about putting it together. And we're going to start looking at the mechanism for cytotoxic T cells first, then we'll put it all together. Okay. So in step 1, we're going to have a virus, and the virus invades a host cell. So here's the virus, and you can see here the virus, how it's invading the host cell. When the virus invades the host cell, the virus will take over the genetic machinery, right? They'll take over the cell, and the virus will produce viral proteins, and those viral proteins will become fragmented. Notice on this cell here, we have these self-antigens, which are MHC molecules. Those are already on the cell surface, just hanging out, waiting there. After that virus has invaded the cell and it's produced its proteins, and the proteins have been fragmented, now we can have your MHC molecule present the antigen on the surface of the cell. We're talking about cytotoxic T cells here that are going to have to bind to this viral antigen. So what class MHC molecule should be binding to this antigen and presenting it to the cytotoxic T cell? Hopefully you said class 1. Class 1 MHCs will present antigens to cytotoxic T cells. So the next step then is for the cytotoxic T cell to come in and bind to the antigen presenting cell. So here's your cytotoxic T cell, and it binds to the antigen. So what this slide does not show is the protein that's going to ensure or enhance the binding of the cytotoxic T cell to the virally invaded host cell. What protein is going to enhance this binding? Remember that your cell, we're dealing with cytotoxic T cells. So this protein that's enhancing the binding must be a CD8 protein. When the cytotoxic T cell, and this is something new, so it's very important to pay attention, when the cytotoxic T cell recognizes and binds to the class 1, okay, and then you get the CD8 enhancing the binding. At the same time that this happens, at the time of your cytotoxic T cell binding to the antigen, the cytotoxic T cell is going to receive a signal from the helper T cell. This signal is in the form of a chemical messenger called interleukin 2. Once we have this occurring, okay, and we'll talk more about this helper T and where this IL-2 thing came from here in just a moment, but let's talk about what the cytotoxic T cell does. We know that cytotoxic T cells, think about the name, cytotoxic. Cyto is cell, toxic, kill. So we want to kill the virally invaded cell. So how does that happen? Well, the cytotoxic T cell is going to release chemicals called perforins. And you already know that natural killer cells release perforins as well. And perforins will create a pore or a hole, right, in the membrane of the virally infected cell. So once that pore gets created, then we know this increases the permeability of the cell to water and ions, and the cell will lyse. Now, just to ensure that this cell is killed, something else happens. The cytotoxic T cell not only releases perforins, also releases a chemical called fragmentins. So this is a chemical that enters the cell via the pore that was created by the perforins. What the fragmentins do when they get into this cell is that they induce apoptosis, programmed cell death. They tell that cell to commit suicide. So here we have sort of a backup system, right? If the perforins that created the pore don't get the cell lysed very quickly, well, you can also rely on apoptosis or programmed cell death, and that cell will kill itself. So now let's put all of this information together. You have a cell, it's been infected or invaded by a virus. We are going to begin with a macrophage. All right, so remember what macrophages do. Macrophages engulf. So they, in this case, have engulfed viral fragmented proteins. So remember, this macrophage could also be a dendritic cell. Okay, it could be other cell types because guess what? You find class 2 MHC molecules associated with them. So what other cell types could this be? go back and review what class 2 MHC molecules are associated with. So when the macrophage engulfs these virally fragmented proteins, then the class 2 MHC will present the antigen. Remember, the antigen is the fragmented protein to the helper T cell, because remember, it's a helper T cell that always recognizes a class 2. So once the class 2 presents the antigen to the helper T cell, at this moment, when they bind, so here we have binding, and you're also going to be using a CD4 to enhance the binding, just like we talked about earlier. So now once this happens, your macrophage will secrete a chemical messenger. It's going to secrete interleukin 1. And when macrophages secrete interleukin 1, that's going to cause a couple things to happen. That's going to cause your helper T cells to proliferate. And when they proliferate, they're going to proliferate into IL-2 secreting helper T cells, and they're going to proliferate into memory cells. So the IL-2 helper T cells go on and they're going to secrete IL-2, interleukin 2, this chemical messenger. Memory cells, we know what memory cells do. Memory cells are going to sit there and hang out until you get exposed again to the same viral antigen. All right, so now focusing on this IL-2 secreting helper T cell. So IL-2 then, once it gets secreted, that's going to cause, and if you think back a few slides to part 5A, you think about the functions of secreting IL-2. One of those functions is to cause the B cells to proliferate. So we activate the B cells, and when we activate the B cells, they can go on to produce antibodies and memory cells. What IL-2 also does is it activates cytotoxic T cells. So here we are here, and we've activated cytotoxic T cells. And when they're activated, now they can bind to a class 1 MHC molecule. So remember, class 1s are on the surface of all nucleated cells, and they're presenting some antigen to the cytotoxic T cell. And so what we need to do here is we need to kill the infected cell. So we know how cytotoxic T cells do that. Cytotoxic T cells will release perforins, and also fragmentins, right? You can see both of them being released by a cytotoxic T cell. That will create, the perforins create the pore, the fragmentins enter through the pore, the perforins can cause lysis of the cell, and fragmentins can cause apoptosis. Now, cytotoxic T cells can then go on and kill other cells as well. So their job is not done once they've caused the death of this cell. So again, here's a nice little flowchart that puts all of this information together for you. So once again, now what we have done is we've gone through the innate immune response. We've covered the adaptive immune response, which is the one that has the memory, right? And the one that's very specific to certain microbes or certain pathogens or tumor cells. We've kind of ended here at a point where we've put all this information together and how the innate response and how the adaptive response actually have to work together in order to do this. So the next, or the last, which is going to be part 6 video, is on the immune responses in health and disease.